Welcome to today's lecture on the fascinating topic of DNA replication. In this script, we will dive into the intricate details of the molecular machinery involved in this crucial biological process. DNA replication is a fundamental process that allows cells to produce identical copies of their genetic material, ensuring accurate transmission of genetic information from one generation to the next. Through this lecture, we will explore the various enzymes, proteins, and mechanisms that contribute to DNA replication. So, let's start our journey by understanding the molecular machinery of DNA replication. DNA replication is a complex process that involves multiple proteins and enzymes working together. At the core of this machinery is a large protein complex called the replisome, which coordinates the replication process. The replisome consists of several key components, including DNA helicases, DNA polymerases, and DNA primases. These proteins are responsible for unwinding the double helix, synthesizing new DNA strands, and initiating DNA replication. Additionally, other proteins such as single-strand binding proteins, toposomerases, and DNA ligases contribute to the replication process. DNA helicases are enzymes that play a crucial role in DNA replication by unwinding the double helix. They utilize energy from ATP hydrolysis to break the hydrogen bonds between the base pairs and separate the two DNA strands. By creating a replication fork, DNA helicases provide access to the DNA template for replication enzymes, such as DNA polymerases. DNA polymerases are enzymes responsible for synthesizing new DNA strands during replication. They utilize the existing DNA strands as templates and add complementary nucleotides sequentially to synthesize the new strands. DNA polymerases also possess proofreading capabilities to correct errors made during synthesis, ensuring the fidelity of DNA replication. DNA primases are specialized enzymes that synthesize short RNA primers necessary for DNA replication. Primers provide the starting point for DNA polymerases to initiate strand synthesis. DNA primases ensure that DNA polymerases have a free 3O group to which they can add nucleotides. Once the RNA primers are synthesized, DNA polymerases can extend them by adding DNA nucleotides to form the complete DNA strands. During DNA replication, the two DNA strands are synthesized in different directions due to the antiparallel nature of the DNA double helix. The leading strand is synthesized continuously in the 5-3 direction, following the replication fork. In contrast, the lagging strand is synthesized discontinuously in short fragments called Okazaki fragments. Okazaki fragments are short DNA fragments synthesized on the lagging strand during DNA replication. These fragments are initiated by RNA primers and synthesized in the 5-3 direction away from the replication fork. Once the Okazaki fragments are synthesized, they are joined together by DNA ligase to form a continuous DNA strand. Telomeres are repetitive DNA sequences found at the ends of chromosomes. They protect the genetic material from degradation and prevent the loss of crucial coding regions during replication. Telomerase is an enzyme that maintains telomere length by adding repetitive DNA sequences to the ends of chromosomes. Telomerase activity is essential for the longevity of cells, and its dysregulation has been implicated in aging and cancer. DNA replication is a highly accurate process, thanks to the fidelity mechanisms employed by DNA polymerases. These mechanisms include proofreading during synthesis and the ability of DNA polymerases to remove and replace incorrect nucleotides. Additionally, other DNA repair pathways actively correct errors that may occur during replication, ensuring the integrity of the genetic material. Despite the fidelity mechanisms, DNA replication can still encounter various types of errors and damage. DNA damage can arise from exogenous factors such as radiation or mutagenic chemicals, as well as endogenous factors like reactive oxygen species. Cells employ several DNA repair mechanisms, including base excision repair, nucleotide excision repair, and mismatch repair, to maintain the integrity of the genome and prevent the accumulation of mutations. DNA replication is tightly regulated to ensure accurate and timely duplication of the genome. Cell cycle checkpoints monitor the progress of DNA replication and coordinate it with other cellular processes. These checkpoints assess DNA integrity, DNA synthesis completion, and replication fork stability. If any abnormalities are detected, the checkpoints halt the cell cycle progression to allow for DNA repair or, in severe cases, induce cell death to prevent the propagation of damaged DNA. DNA replication begins at specific sites called replication origins. Replication origins are characterized by specific DNA sequences and associated proteins that recruit the replication machinery. The initiation of replication involves the assembly of the replisome and the loading of helicases, polymerases, and primases onto the DNA. Initiators and licensing factors tightly control the formation of replication forks, ensuring replication occurs only once per cell cycle. DNA replication is a crucial step in both mitosis and meiosis. Mitosis is the process of cell division that produces two identical daughter cells, each with the same genetic material as the parent cell. Meiosis, on the other hand, is a specialized form of cell division that produces gametes, sperm and eggs, with half the number of chromosomes as the parent cell. Proper DNA replication is vital for the accurate segregation of chromosomes during these processes. In conclusion, DNA replication is a complex and highly regulated process that ensures accurate transmission of genetic information. The molecular machinery involved, including DNA helicases, polymerases, and primases, work together to unwind the DNA, synthesize new strands, and maintain genome integrity. The fidelity mechanisms, DNA repair pathways, and cell cycle checkpoints further enhance the accuracy and efficiency of DNA replication. Understanding the intricacies of DNA replication is essential for comprehending various biological processes and diseases related to DNA replication defects.